I am Professor Himanshu Kumar, doing teaching and research at Indian Institute of Science Education and Research, Bhopal, Laboratory of Immunology and Infectious Diseases, Department of Biological Sciences. I work with host virus interaction using RNA viruses such as influenza virus, chikungunya virus, dengue virus, and recently I have started working with the Nipah virus. During my research career, I have created several transgenic mice, and in transgenic mice I have created conditional knockout mice, conventional knockout mice using homologous recombination method, and I have also learnt about the CRISPR-Cas9 mediated transgenesis. So, this 4-week course entitled Animal Transgenesis Principle and Methods will basically give an overview about this topic. So, normal animal when we do some genetic manipulation, then this condition we call it as a transgenic, or those organism we can call it as a transgenic organism. So, we will discuss about or I will introduce about the transgenesis. I will also talk about the history of transgenesis. I will talk about need for transgenesis, why transgenesis is needed. I will discuss about model organism, 3R philosophy. 3R stand for replace, reduce and redefine. This philosophy is one has to understand before going for the transgenesis. Then I will talk about the principle of transgenesis, and you may know that this transgenesis needs extensive expertise in molecular biology and cell culture techniques. Therefore, I will give you more detail about the cell culture technique, and over there I will introduce again about the cell culture, and then I will discuss about the history of cell culture, how this technique evolved over the time. Then I will talk about the cell culture types, then basic requirements for cell culture, and then cell culture media and challenges associated with the cell culture. So, this is very much important and foundation for the before going for transgenesis or making transgenic animal. Then I will talk about the steps for generation of transgenic mouse, and this includes manipulation of gene of interest. Then carrier for transgenesis, sometimes people use targeting vector, sometimes people use some viruses. Then I will talk about the methods for transgene introduction in target cell, and then I will talk about the methods for transgene integration in the target cell. There are two major methods, that is homologous recombination and CRISPR-Cas9 mediated transgenesis. Then I will discuss about the screening of transgenic cells, and then I will discuss about the generation of chimeric mice using this transgenic cells, and eventually we will conclude about the generation of transgenic mice. Then I will discuss in more detail about the generation of transgenic mice using homologous recombination method. Over there I will take you to each step in more detail, and then I will discuss in more detail about the conventional transgenic mice, generation of conventional transgenic mice and conditional transgenic mice, and then its application in the research. Then eventually I will move to the generation of transgenic mouse by CRISPR-Cas9 method. This is one of very interesting discovery in recent past. So, I will talk about the discovery of CRISPR-Cas9 system, which is originally discovered in the bacterial cell, and then I will talk about the detailed steps for generation of various transgenic mice, that includes conventional knockout mice or conventional transgenic mice or conditional transgenic mice and its application in research, and then I will also discuss about how we can manipulate this CRISPR-Cas9 system, particularly the enzyme Cas9. So, we will discuss about the Cas9 variants and its application in research, and finally I will finish this course with the translational aspect of CRISPR-Cas9, where I will discuss about how this CRISPR-Cas9 can be exploited in diagnosis, in correction of genetic diseases, treatment of blood cancer, treatment of cardiovascular disease, and treatment of infectious diseases, particularly the HIV infection or AIDS. So, with this I will complete this 4-week course. I hope you will enjoy. Please take this course and try to learn more about the transgenesis and its application in biomedical sciences. Thank you.